And what I wanted to do in this short time that we've got for discussion is just to sort of bring up some of those key challenges that we're facing, and to pick your brains a little bit about what you think the solutions might be, and how we can kind of overcome them together as a community. So I'm going to suggest 3 main challenges, and feel free to absolutely disagree with me, or feel free to suggest ones that you think are more important. Anything like that would be good. So for me, I was thinking from the talks that we've heard, probably one of the major challenges is really public understanding of science. So thinking about this sort of information challenge that we face, and George brought that up in his talk, and Ken as well in his, how do we really kind of address some of those issues? Because this is outside our comfort zone as scientists sometimes. So we need to think about that, because that's a pretty important issue. I think another one is really thinking about global collaboration. That's something that we are used to as scientists, but of course there are challenges that we're now facing. Are there communities within the scientific arena that we're not working with that we should be? There's obviously geography around that as well. Where are those challenges that we're facing? And obviously politics feeds into that too, which we're also sometimes uncomfortable about. And then the third topic is one that I think we haven't really addressed. I think it's been skirted over, but it would be good to dive into. And that is inequality in research. So particularly just thinking about low-income countries, low-middle-income countries, and the challenges that we face around that, particularly with sourcing reagents, costs, logistics, bureaucracy, samples, and really attributing data to the right teams who are actually doing the work, and ensuring that the quality of science that we know is being carried out in those countries is communicated appropriately and attributed appropriately as well, to give full value and credit to those teams. So that's 3 topics. We haven't got all day, so we want to try and crack on with that. And I'm just going to, maybe we'll go in reverse order, backwards. We'll think about these inequality topics first. Nilika, I know you've got a kind of an interest in that. Would you like to kind of comment a little bit more about that, or if you think that you disagree with me, please clarify.
Yes. So that is personal experience. I am from LMIC. I've been working in LMIC for, I don't know, since 2008, since I went back to Sri Lanka. And it's, of course, many challenges. So as an example, and it's not just our problem. So Sri Lanka is one example, but I know many low-income countries and low-middle LMICs face the same challenge. And it's not our problem, but it's everybody's problem. I'll give a good example about the chikungunya outbreak this year. So last December, we realized that there could be a chikungunya outbreak because the clinical features were different from dengue. So towards the latter part of December, Christmas holiday sort of, so first week of December, ordered the primers for diagnostics and sequencing, first week of January. Also put out the IRB approval for all the research to be done on chikungunya, although not worked on chikungunya, starting from innate immunity, antibodies, T cells, sequencing, everything was put in January itself. So by April, we still hadn't got the primers, you see? And people were asking, do you have chikungunya or not? Okay, so, I mean, yes, clinically, yes, we do. But, how do we know? So then one of our collaborators, Ruki, who is from Asia PGI, she came to Sri Lanka in April, so she hand-carried the primers. And when we actually got the primers, it was end of May for sequencing. By that time, we had already sequenced and put out a preprint. So without somebody coming in, like Ruki from Singapore coming in with them, even by May, we would not be able to do it. So now chikungunya is not a virus that spreads so rapidly, but what if it was something else? So it's not just about Sri Lanka, but, so this becomes everybody's problem because it's a threat. And I think that's something, so as far as I'm concerned, I did everything right. We ordered, we took IRB approval, I can't do anything more than that. So, yeah, but this is the situation in many of the LMICs. Yes.
Jean, I don't know if you have any input on that.
I completely agree. And I think one of the problems is there's not agile funding systems. So when you want to have money flow to get things to, it's hard to get money to locations. And so, and that's even in the US. I mean, there's government funding, philanthropic funding, industry funding, but it's never consistent and it's never at the right time. And so that's going to be a problem globally. We're going to have to find ways to get reagents to locations quickly and allow that to happen. And of course, still have regional centers that can help out as well. So.
George, any thoughts?
I totally agree with you with the first two issues or challenges for your conclusion. I cannot agree with you with the third one. This is what we are talking about. Think about it, the research. Historically, all those revolutionary research is done by someone. We only have one Newton. We only have one Einstein, quorum science, right? If we have all the people there, Einstein and Newton, can you imagine what will happen? So I do not agree with you. Equity, equability would share all the agents altogether for, give everybody for the research. Research, science, only for a small group. We don't want to waste of the money to give someone who just, use your life claiming they are scientists. They are not really doing science. So, any breakthrough for science itself. So I would agree with you for the equity and equality, for the accessibility for any public public service, or any shared wealth. That's you need equity, equability. But we do need equity, equality under the so-called, the US now, the DEI, whatever, for the scientists, for science. Scientists, we only need a small group who want to devote their life for the discovery. Thank you. That's my opinion. Anyway, could be wrong.
I guess I just trying to understand a little bit about what you were saying there. So do you think that, I think we agree that there are certain places which are, that the world is unequal, for sure. But from a point of view of ensuring that the groups that need the funding, that are well qualified to have that funding, but the challenge is at the moment that even though they are qualified to have the funding, or it's not even about that. It's just sometimes logistics that's the challenge. It's not even to do with money all the time.
If you think, I think, though the scientists or the professionals, we are not happy or unhappy with Robert Jr. and Trump administration for this. But think about that we have so many scientists, to be honest, because I was the vice president of the National Science Foundation of China for 5 years. So if you go and scrutinize some of the projects, it's just repeated. It's not necessary to do. So this is why when we are talking about equity, equality, what I mean, for the scientific breakthrough, we need more people. So this is, from that point, I do agree with Trump. We need more engineers. We need more people, to really work. So like we need pro-immune company. We don't need some of the small groups to work, consume your, reagents, whatsoever. So this is what I mean. So when you are talking about, I think competition is more important than collaboration in science. Collaboration is more important in politics, in social science. But for science, we need competition.
Okay, this is a good.
If you can compete, gather funds, you are great. Excellent. If you can't, well, okay. So this is what I mean.
You, Cho, please, over to you.
In my opinion, actually somehow I agree with you, somehow I agree with you. So in my opinion, there are two different kinds of science. In one kind of science, it's just like Einstein, so like that the revealing some of the nature's law, and then in that way, the one or two outstanding people are leading the science. But the other, second kind of science, like the preparedness, the emerging virus, in this case, just brightness is not the one of the part, not the all. So in the case of this the practical issue, dealing with the practical issue, those kind of science, so we need not only outstanding, but also not just one or two people. So we need really the global collaboration. So I think that we can discuss about that science, two different kind of science, in my opinion.
Thank you.
Yeah, I mean, I had the privilege of chairing a grant review committee for Wellcome Trust recently on Zika and dengue, and okay. Sorry, yeah, and I think one of the things that I liked about it was that so first of all, this was not discovery science. This was trying to understand prevalence of a medical problem, which is probably more widespread in the developing world than in places like Europe and North America. And so part of the collaboration was that they required a collaboration between an established lab that could lead the science, but with scientists from places where there is endemic problem. And I thought that was a very happy marriage, right? Where you're getting the top scientists from institutes where they can bring some really sophisticated tools, but you're collaborating with people on the ground where the problem is happening. I think maybe we need models like that, but that is to address in a specific question on, what's the crosstalk between dengue and Zika, and so you have to be where the problem is, where you have to be where the virus is. Whereas it's quite different if it's got to do with discovery science, like the next CRISPR or the next revolutionary DNA technology. That you just want to fund the best ideas, and you don't care where that is from. But maybe since I might have one more added issue. To me, when you said equity, to me the issue is this, right? That, for instance, even with COVID-19, some of the clinical trials were done in places where you have a lot of disease, like South Africa. And yet, when the vaccine was licensed, South Africa wasn't online to receive it. To me, that was unacceptable, right? If we're going to take advantage of places where there is disease, then there has to be some, equity in terms of the access of the product when it first comes out. And maybe that's a fault of our society where we have left in some ways in product development and all, like vaccines, to corporations. And corporations have a duty to report back to their shareholders. After all, that's where the capital came from. So you cannot blame them for not selling the vaccines cheaply back to South Africa, because ultimately J&J still had to report to their shareholders. But I think what may be needed is somewhere where there's this halfway house, because back when we had polio vaccine, when it first came out, there was no company that sold it. It was actually manufactured in country, right? So the Albert Sabin and others who discovered the vaccine gave out the seeds, and the manufacturing was done in country. The problem with that was that there was no innovation, because once you've built the factory, then that's all you can produce. So maybe what we need is somewhere in between, right? Where you have the innovation, but you also have some in-country or in-region production, so that, in places that we don't have this equity where the first comers are those who have the money, and if you don't have the money, well, you just stand in the queue.
Yeah, well, I think a good example of that also was the Oxford AstraZeneca vaccine, where there was obviously great collaboration with the Indian Serum Institute, and actually that was designed right from the very beginning, and actually that was a prerequisite for the university wanting the deal that they had with AstraZeneca. So I think that was quite nice.
Yeah, I just wanted to add to the two levels of science. So yes, discovery science, that's very competitive. That's a different type of thing, I think. But when you take something like outbreaks, I think, yes, you need to answer the very, high level, that sort of question. But sometimes doing very, very simple, basic type of research can come up with very quick answers, repurposing drugs, which can actually make a difference to millions of people's lives. So something like chikungunya, let's say chikungunya, dengue, I mean, we don't have treatment. So sometimes you don't need to make like rocket, like making level of science. Sometimes you just need to answer some basic questions, do some very simple research, because you want rapid solutions to do something like that. And that would could actually impact millions and millions of people's lives. So, so this is why I think there are two different levels of research. And I think, yes, those really groundbreaking research questions, Einstein and Isaac type of research questions, yes, competitiveness, all that is important. But at the same time, we are doing science to benefit mankind. And I think sometimes you don't need rocket science to benefit mankind. And at such instances, you do need to deploy rapid tools to bring solutions. Yes.
I was going to say we probably do need rocket science if we have to think about planetary health, but there we go. So what about, in regards to the, I mean, we're sort of talking about collaborations in that context of science. Maybe let's talk about that, because how practically can we actually achieve some of those resolutions, I guess, to these challenges? Because it always is like a bit of an amorphous concept, and we just talk about, we need more collaboration. Practically, particularly given where we are headed at the moment in near term in regards to the world's politics, it's a challenging time to have those global connections made. Where in the past, since the 1940s, it's been very much right. We can work together, WHO, those sorts of really important bodies. And so what as grassroots scientists, and maybe George is probably now slightly different level to that for sure, but we're all scientists, and I think from that point of view, what can we do to foster those collaborations in a practical way? So any thoughts? I know it's not an easy one.
I think I'm going to go a little off from that, but I kind of agree with George, and it's something that we tend to, we talk about competition and collaboration. It's almost like a ratio, and science is full of history. And if you go back to Koch and to Pasteur, that was both a national and a personal competition that drove a lot of science. And so you want a little bit of competition, but you want it to be healthy, but you also want collaboration at the same time, and maybe pockets of the right type of collaboration. I mean, for me personally, it's one of the problems I struggle with is fragmentation of the inability to have a basic science understanding, a person understand that, by the time you get to clinical, that's going to be almost impossible to do. But at the same time, having someone who's at that clinical side seeing the value of something really basic, and that transition and it's very fragmented. So I think George might have a comment on competition.
I also comment for Dr. Shin. So when you're talking about two levels of science, that's really touch the base of the definition of science. So if we are talking about science and the research, we want to know the unknown. If you want to know the unknown, you really want to give the money to someone who can really do that. That's the discovery. If you give the money, you put the money to so many people because of the equity and equality, then by the end, there will nothing happen. Like you said, older they call, a pastor, that's kind of a personal competition. Nowadays, because of the so-called DEI, all the government and then the funding body want to interfere with this. If your interference goes the wrong way, that's really, will be the totally different results. So for me, I think if you want to do the research, define what is science. Science is the discovery. It's for something new. You want to make a comment? Come on. Come on. Come on. You sit here.
I think I agree with you, but it's too short-sighted. I think this is the unfair word. I think long-termly, this EID thing is really important, because I think as a Chinese being in Oxford for so long, I believe I just have the same brain as any man and any British people, but they do, because the language, the look, your skin color, you miss lots of opportunities. But with the implementation of this EID idea, personally, I benefited a lot. You know Andrew. Andrew is one of these great leaders who don't have these filtered eyes. We both benefited. You actually benefited from Andrew's EID idea. It's a long-term effect. Now, Andrew, see what we have got. As the result of the EID policy, we got George Gao.
Thank you, Tao.
Tao partly agree with you. So you are addressing more on the training. Training and research, they are different. If you are running a training program, you want to give to the young generation, very good chance and an equal chance. But I'm talking about the size. So this is why I don't think we have time to discuss about what science is, what technology is, what engineering is. So we need to really think about science, technology, engineering. And now Tao touched the base about the training. For the training, it's different. If you're a professor in the university, like here, National University of Singapore, so you really need to train, good students and good future generation. Okay, thank you.
So maybe it's better to go back to the original topic. So actually Jeremy was talking about the global collaboration, some practical issue. So when we talk about the collaboration in biology, so just we can think about the open mind or attitude or like the collaboration. But more practical issue is the standardized technique. So actually, so in physics, for example, so if I measure 1 meter, then his lab also 1 meter. We can just communicate with the same unit. But in the biology, immunology, so for example, he cannot use my lab's flow cytometry data just directly. So we cannot directly share that because the different machine, different voltage in that way. So in immunology, we try to make some common language, common unit, like the standardized SOP or like that. So for example, in my lab, so it doesn't matter for AMOS, tetramer, within my lab. But we need to try to the interchangeable some information. So I think that in biology, immunology, one starting point is the single cell analysis. For example, in single cell, so I can merge his single cell data with my single cell data, just batch correction. So in that way, single cell is one of the good way, but in the flow also, so we need to develop some common language that you need. That's one of the practical issues in immunology, especially T cell study, for example. Yeah.
It's also similar to maybe some of the animal.
But I mean, we all trying now to do this SOP. We all should use the same assay, but this blocks innovation. I mean, if I'm doing in my assay, I do my standard protocol, and now I see another marker is actually much more important, and now I can't add it because another lab is not adding it. No, I don't know. I don't think this idea of we all, I mean, in a way, what we are doing every day, we are changing our assay, isn't it? I mean, most of us don't use standard SOP. We are not a hospital.
Okay, just a quick one. So in terms of access and so on, I mean, there's the question of what type of science to do, but practically in immunology, you need to have access to the patients, the samples, and they're clearly there's a geographical distribution to them. And in tropical infectious diseases, I mean, that distribution may be in lower middle income countries. So, you need to study those people, those samples, and so on. And it seems to me that, like in your case, when you have an outbreak, you need to study the people who are there, who are having the outbreak, to have the latest insights. And, from a practical point of view, if that's delayed and so on, that's not helpful. I mean, and who's ultimately doing the science perhaps doesn't matter, but it might as well be the people who are on site in the country. So do you know why your primers didn't arrive? I mean.
So just to let you know, not just in Sri Lanka, many countries, when you put an order, it takes a long time to get things because of the very small number of people doing research in the country. So even if it's so, the companies put things together and order things so that the shipping costs are less and all that. So, and then when it arrives, there's a lot of bureaucracy in clearing, custom clearance. And so if you take the same, let's say, a QIAGEN RNA extraction kit, if from Oxford, because I know the price of RNA extraction kit in Oxford versus Sri Lanka, it's like twice the price in Sri Lanka than in Oxford. So even if you have the same amount of funds, you can do half amount the work because it's twice the cost than in Oxford. So, and all the reagents are like that. Because you have the shipping costs, the very small critical mass doing research in many LMICs. So when you compare to, I can compare with Oxford because I work there. It's twice the price. Even a flow cytometry antibody, every single thing is twice the price. So that really puts everything at a disadvantage.
From my point of view, it's partly solving some of these practical problems. I mean, we ship direct into, we like to not use distributors. So we ship direct to countries. Sometimes that's problematic. Shipping direct to China is difficult, and clearing customs. Brazil is also very difficult, but I think Sri Lanka, I think, is not so difficult. I mean, a lot of people in those countries order through distributors. So I think, sometimes it's a question of, are there other ways around those really practical issues, just getting, some basic resources into place. And, well, actually, because of the funding cuts in the US, we've bought some reagents for some of our friends in the FDA so they can continue some of their work. So it's not just low and middle income countries who are sometimes impeded by very practical problems. And, I think maybe the first step is to try and work out how to unpick some of those.
Yeah, thank you, Nick. Just probably so we can come to an end, I want to focus on the last topic of the challenge of information and communication to those who may be not fully understanding the detailed technical aspects of vaccinology or immunology or infectious disease science. One of the things that I was chatting to Nicola earlier about was just, even as young scientists in training, one thing that I was exposed to as a young scientist was basically, as part of my PhD, having the opportunity to do some schools work and actually going into a secondary school and having to kind of communicate complex science topics. So not things on their curriculum, but what you're doing in your PhD, but in a more simple way. And that was actually really helpful. I think it was helpful for them because they were enthused in a way that they hadn't been before. It's useful for me to kind of put my language and frame it in a new way. So what can we do with social media now being what it is? It's a very different world from when we were younger. But what can we now do to counter the challenges of regards to information? Hang on, you've got any comments?
I don't know about communicating with the public is one challenge, but actually, I think to me, there's a closer challenge to what we're trying to do, right? And that's to, like rapid translation, 100-day vaccine, and all that. How many of you are from Health Sciences Authority, HSA? See my point exactly. No one. And that's the regulator. Yeah. And so we've been talking about T cells and cell-mediated immunity being important, but the regulator does not understand T cell. So we still have to fall back on antibody.
We wanted them to come, but...
Right. And closer to and that's really close to home because it's really what we're trying to do, and yet there is no communication. And when we do need to communicate with them, it's in a crisis, and they don't understand the science. So there's nothing you can do, And that to me is a more fundamental problem. I mean, clearly communicating science to the public is also important to overcome misinformation. But ultimately, the public also looks to who licenses, who approved this product. And if the authority that approved the product cannot explain why they approved it, then we're back to square one.
I recently, I believe the author's name is Max Brooks. He wrote the World War Z book and then became a movie. He actually gave a talk recently, and he was mentioning that scientists should not communicate to the public. He was very clear about it, and he says it's even more problematic now because the social media is so segmented that we can't work in all those areas. We can't think about how one group of people, which is really a tribe, and another tribe, and a third tribe, how they're thinking, and your job is to do the science. And again, let communicators that know how to do that properly do their job. But we're not interacting with those communicators. There's people that can do that kind of work.
So Yuchol and I were having a quick chat earlier, and he gave a very good example, which actually is a situation where he does have to communicate because he's writing a paper. The question is, how does he write the title of that paper? Because it may get picked up by people and interpreted in the wrong way. Yuchol, do you want to explain what you were thinking?
So actually, I have before talking about that, just I'll answer to Nina's comment, a little short comment. Actually, your comment. So I'm not talking about that we may must use the same protocol, same marker, but so we can try to add new marker, but in each lab, but we need to consensus core marker. So that was my idea. So not the homogeneous. Yeah. Anyway, let's come back to the information and this one. So in my idea, so I have the opinion to you. Actually, during pandemic, I communicate a lot in publics, in YouTube, for example, in Korean, some language. So actually, I was requested by the medical newspaper. So their channel, I explain about the theory, principle of vaccination, what's T cell, what's antibody, and then how we can do the PCR for SARS-CoV-2 in that way. Actually, the series of like 20 or like the YouTube. But meanwhile, I found that, so this one is also very pleasant experience for me. And then, so actually there are some, the comment on my YouTube. Really, I found that many people, they learn about T cell from my lecture. But it is very important. How easy content, and then we need to develop better idea. So even after such kind of YouTube activity, so recently I was requested from some middle school or elementary school. So would you the lecture about some T cell or about the antibody? So even I made my own I don't know the English name, the small the character with the handmade one. Yeah, in that way, I can do some play. The B cell, T cell, in that way. So I think that this is very active engagement to the public communication. So not every scientist, but so we think about more active engagement if you are enjoying such kind of activity.
Actually, we're in a company of someone who produced a manga cartoon on immunology, right? Ken Ishii.
Okay, so we have a manga called Cells at Work. Just check it out. You can Google it now. Cells at Work is the best textbook for immunology. And we are currently working on this new one with the drug and vaccines. I have never had a retracted paper in my life, but I had to retract one of the manga story based on the fans' claim that there was a wrong description in scientific manner. So I had to retract that manga, and I gave an apology letter. So people are watching you, whether you are doing good science or not. And based on your science background, they trust you or not. And now there are many channels. You can go YouTube, but also we have to have also serious communication with policymakers and politicians. But scientists has a culture that they are, if you talk to politician or media, you are kind of second-class scientist. It used to be like this, but not anymore after COVID-19. So that's why we are. But please try to look at Cells at Work.
Good plug. Thank you, Ken. Thank you very much. I mean, actually, in the UK, we were, amidst the pandemic, was not in some respects, was the country's finest hour recently in regards to science, but politically not. And I think actually the great British public were probably quite glad of hearing some of the scientists giving their talk in a non-jazzy way. We won't talk about that anymore. But yeah, it was one of those things. Actually, having the scientists, I think, gave people a little bit more comfort that there was somebody who knew a little bit about what was going on. We're running out of time, so I was just wondering if there were any sort of final burning comments that any of the panel wanted to kind of bring up that we felt that we hadn't covered. No burning points. Well, on that, sorry, no, Jean, you.
No, just ask them.
Anything from the floor is a dangerous thing to ask. No, we're okay. We're okay, good. Well, it just remains me to thank the panelists for their contributions. Thank you very much indeed.